I mean, maybe flow to form or something like that. If anybody has some problem in a particular topic, I can maybe go
yes,
back
sir.
to it.
That's right. The thing is that suppose for one particular experiment, like suppose for example we are like studying the experiment of that urine levels of 8-oxoglutamate 8-oxothymine,
Guanine.
8-oxoguanine, sorry, 8-oxoguanine. Suppose now the thing is that there are 2 or 3 graphs. So we are understanding that okay, so because of this problem this is happening, so the DNA repair system is not actually working well, uh, and since reactive oxygen species accumulates, so that causes aging. But the thing is that, like suppose for 2 3 graphs, they belong to the same experiment, and they are actually correlated with each other. So we are not quite getting into the correlation between all those graphs. So suppose means this is just an example of the some of the problems we are facing actually while understanding and studying this subject. So that's it.
Let's see how it goes. I really hope it, I mean, you people try to understand. Maybe the assignment will make things a little bit more clear.
Maybe I guess the research papers, reading the research papers will do a help, I guess.
Yes, because I mean, the way you understand, the way I understand might be different. So let's see. It's a learning phase for all of us, and I hope everything goes on well. Starting off with the first slide, I mean, you just said about evidences about linking reactive oxygen species and aging. You said you have, I mean, given a very nice flow chart. How reactive oxygen species, they cause DNA damage and whatever happens. So these two papers are involved, which we'll be studying mostly, and we will be studying about Hayflick's limit. So anybody knows anything about Hayflick's limit yet?
No, sir. No, sir.
So you have not studied animal biotechnology yet. You will be taught about it. We'll be talking about it. So the first paper, which talks about extension of Drosophila lifespan by SOD1 overexpression. So essentially, in this paper, the authors, they took they took SOD null mutants of Drosophila. So SOD null mutants means they cannot produce SOD by themselves, right? And they saw that these kind of Drosophila, these flies, they died very soon. But when human SOD1 was supplemented to these SOD null mutants, so their lifespan was recovered. Is that clear?
Yes, sir.
So in the y-axis, you see the survivorship percentage, x-axis, you see the adult age. So whenever we, give SOD1, HS is actually human SOD1. When this is supplemented into the SOD null mutants, just give me a moment. At that time, their lifespan is increased. If there is similarly, we see the same two graphs for SOD1 and SOD2. So here also we observe that SOD1 overexpression extends the lifespan of Drosophila more than that of SOD2. So you can go through it. The papers have been given. So SOD null
That
mutants.
means we are like introducing the human SOD gene into the Drosophila.
Yes.
Achha, okay.
So what happens? SOD null mutants of Drosophila, they are short-lived. And the transgenic Drosophila, why is it transgenic? Because it is not expressing its own gene. It is a gene from humans. So transgenic version of Drosophila, which expresses human SOD1, there we observe an extension of lifespan by 40%. Is that clear?
Yes, sir.
Yeah. Very less attendance today. Anyway, uh, if we see some papers regarding the correlation of lifespan with specific metabolic rate. So here we see that we have already read few papers, right, where superoxide dismutase activity levels were seen in different tissues.
Yes, sir.
So we see that SOD levels, they decrease with age, that we have seen. Another thing that is very interesting is that okay, it's the graph is not given here, that the rate of SOD activity to specific metabolic rate was found to increase with increasing maximum lifespan potential. So what does this mean? That if the maximum lifespan of an organism is more, then the SOD activity will also be more in that organism. Is that clear so far?
Yes, sir. Mm-hmm.
Just for the sake of everybody, if this is, let's say, SOD activity, normalizing it with metabolic rate, and if this is the lifespan of an organism, how do you expect the graph to be?
It will be an increasing curve.
Increasing curve like this?
Yes, sir. But sir,
Yeah, you.
মানে, here it is written the ratio of SOD activity to specific metabolic rate was found to increase with increasing lifespan. So, uh, shouldn't be like the reverse thing, like lifespan is increasing because of SOD activity, like SOD activity per unit
This
metabolic
is a
rate
correlation plot. This is a correlation plot, right?
correlation plot.
You are
Okay.
right, but this
Why
is
will
a
lifespan
correlation plot.
decrease?
Lifespan.
Why will lifespan decrease? Like he said that lifespan should decrease according to this, but why?
No,
Like
not decrease. I am saying SOD activity per unit metabolic rate. This parameter should be in the x-axis and lifespan should be in the y-axis. But actually, the thing is, this is a correlation graph. Like y is a function of x and x is a function of y. That is the thing.
Is that clear, Shinjan, or
no,
you
sir.
need some more explanation?
More
No.
explanation, sir.
Yeah. So what he's trying to say is that if there is, let's say, less SOD2 activity, or there is higher metabolic, I mean, uh, we have seen organisms which have higher metabolic, specific metabolic rate, they have a lower lifespan, right?
Yes, sir.
That we have already seen that, right?
Mhm.
So what trying to understand here is, why are we comparing like lifespan with this? I mean, it should be the other way around. That if the SOD activity is lower, then the lifespan should be lower as well. I mean, you're understanding the point,
Yes, sir.
but I represented it in a different way.
Okay.
Okay, here, anybody would like to state any observations? This was seen in nuclear DNA,
MLS P
maximum lifespan. This is mitochondrial DNA. Any observations?
So since SOD concentration is more profoundly found in mitochondria, that area, that's why we see this hyperbolic slope. But for genomic DNA, it's inside the nucleus and more protected by the nuclear membrane, I guess. That's why we can't see that SOD2 reacting much.
SOD2
Right.
দেখছি না এখানে।
Then
8-oxo guanine আমি দেখছি। 8-oxo
also
guanine with respect to guanine, মানে the ratio of 8-oxo guanine with respect to guanine. So what do we observe? What is the indicator, মানে 8-oxo guanine মানে কি, মানে what does that indicate?
Mutated nucleotide.
মানে কেন
Or
মিউটেশনটা
mutation.
হচ্ছে কেন?
because due to reactive oxygen species.
Right, right, right. You're right. So reactive
Acha,
oxygen
so
species.
due to the accumulation of reactive oxygen species causes increasing levels of 8-oxoguanosine. Now,
Right.
as the means again, it is a bit of correlation thing that as the amount of 8-oxoguanosine is decreasing, the lifespan of that particular organism also increases. Like for example, that means that DNA repair system, or like DNA damage repair system, is more functional in the case of horse as compared to a guinea pig. So that is the thing.
Fine, fine, fine. That's a good
And
observation.
this relation is more linear. But in the case of mitochondrial DNA, since the reactive oxygen species is mainly present in the it is actually generated inside mitochondria, so it is more prone to destroying the mitochondrial DNA as compared to the nuclear DNA. So therefore, it might be said that if the reactive oxygen species is nullified or eliminated in the in the mitochondria itself, the lifespan would increase much more, because since mitochondrial
That
DNA,
is
since because mitochondria is the powerhouse of the cell. So since mitochondria is the powerhouse of the cell, so we can say that if mitochondrial DNA gets damaged, or mitochondria gets damaged in anyhow manner, so the amount of 8-oxo if the amount of 8-oxoguanosine increases, the rate of increase of 8-oxoguanine will be more in the case of mitochondrial DNA, because mitochondrial DNA contains does not contain exons actually, but nuclear DNA contains exons. So the rate of mutation in
I mean, Antik, you have gone through, I mean,
maybe
too far.
maybe
I
maybe
mean,
maybe maybe
I mean, your inference was right, but you have made some hypothesis also. I will not comment anything on that. But
maybe
you were able to make some right inferences. So essentially, what happens in case of mitochondrial DNA? There is higher oxidative damage. Hence, we see a more, I mean, the if you see the R square value, the fit of this curve is better in case of mitochondrial DNA than that of nuclear DNA. So I mean, that is correct. What you said, that ROS induced damage is more in that case of mitochondrial DNA. Whatever you said later, whether rescuing the damage of
since
mitochondrial
we
DNA
know that
would.
by biological evolutionary history, since a eukaryotic cell is formed, because I have studied this very little, but that one prokaryotic cell has engulfed another smaller prokaryotic cell, and then it has formed a
Okay.
mutualistic association with the host cell.
Mhm.
So it will give energy to the host cell, and host cell will provide it nutrients. So that became mitochondria, and that cell became from prokaryotic, it changed to a eukaryotic cell. So means
You're talking about the
evolution.
endosymbiotic origin of life, right?
What
So
is that called? That term I have forgot actually.
Endosymbiotic. Mayuk,
Endosymbiotic, right,
anything?
right.
phagocytosis in
No, 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 no. Phagocytosis is not involved here. We'll be discussing about it later. Anyway,
because the
I mean,
mechanism was similar to that of phagocytosis, that one cell is engulfing another cell, and then
I mean,
inside the system energy is.
what he's trying to say is mitochondria has a prokaryotic origin. It is reported in many papers also. Even if we are eukaryotes, mitochondria has a prokaryotic origin. So essentially, it also has a DNA similar to that of prokaryotes. So what essentially, I mean, scientists hypothesize is that mitochondria was it was kind of a bacteria which has, lived to, I mean, it has evolved to live with humans. I mean, if that makes any sense, I'm not sure. I'm, very novice in evolutionary biology, but these are some of the things that I have read about it. So yeah, prokaryotes, eukaryotes, how prokaryotes have evolved to eukaryotes, those kind of things. So it's not the topic of discussion here, anyway. But it's good that you people are interacting today. Yeah. So we have already discussed about this. Why are we discussing about this again?
Because we have not understood it.
Really? Okay. What is it that you did not understand?
This coenzyme Q bound to mitochondrial this curve, this thing was a bit confusing again for me.
What is coenzyme Q?
Ubiquinone. So, acha, that 384 is part. Right, right. 484
Yeah,
is
you got
384
it.
is thing. Ah,
What is it? Then explain.
the thing is that if the reactive oxygen species concentration increases, then the thing is that wait, I'm explaining. It will be a bit bad, but the thing is uh, the ubiquinone and the ubiquinone and the cyto like the cytochrome complexes, they are active, like, no, sorry, the ubiquinone is, I guess, active, as far as I remember, is active in the 3fe4 state, but in the presence of reactive oxygen species it gets changed to 4fe4s, so
No,
it
no,
cannot.
you are confusing.
No, no.
Again, let me
Yes,
try
yes.
to yeah.
Yes, sir.
So what happens? Ubiquinone are important for relaying of electrons in electron transfer chain. Is that clear?
Yes, sir.
So what has been observed that if there are like more amount of ubiquinones present, then the amount of uh, uh, the peroxide radical, or let's say any reactive oxygen species, is also less. Why so?
Yes.
Why so?
Because they are since they are, no, I don't know.
So
It's
see,
confusing.
if the electrons are not relayed properly through the electron transport chain, then they would be, the electrons are, if they are available, they will form reactive oxygen species. Reactive oxygen species also have an extra electron, right?
Yes, sir.
So if they are not relayed properly, there will be more reactive oxygen species which will be generated. So if there is what you are talking about, I mean, if there is a faulty, if we have talked about rotenone, which in which doesn't allow this coenzyme Q to accept electron. I mean, this is the normal. So this is inhibited by rotenone, right?
Yes, sir.
If you remember. So if we have this inhibitor, then what happens? There is increase in the reactive oxygen species.
Okay, sir. Okay.
So this is what we observed here. Also, you will see correlation with lifespan. So who lives longer, a cow or a mouse? No answer.
Cow,
Yeah. So
because the nanomole of peroxide is less.
I mean, it doesn't have to be any reason why it lives longer, but the thing is that cow lives longer and they also have a higher amount of coenzyme Q associated with their mitochondria. That is an observation. Again, another observation. So the animals which live longer, they have higher amount of coenzyme Q, which is associated with their mitochondria. Is that clear?
Yes, sir.
Okay. I don't have a lot of slides today. Okay, I had a blank slide here. So we will discuss about Hayflick. So nobody knows about Hayflick, I assume. So I need to talk about a little bit about cell
I've
culture
heard
here.
about,
Sorry.
I think I've heard about Wikileaks limit. I
हाँ
think
बताओ. What do you know?
it's the number of times the cell can divide up before it can no longer divide and it will die. Due to telling shortening.
Number of times a cell divides.
Before it can no longer divide, I mean before it dies.
So I mean, is it applicable to all cells?
That I do not know.
Okay, so okay, let's see. We'll discuss a little bit about cell culture. So, what do you know about cell culture? Any idea about animal cells? No idea, nothing.
No, sir.
So
Not
whenever,
studied
let's
it.
say, whenever a human cell is, you have seen a bacterial cell culture, right?
Yes, sir.
So what do? You take the cells, I mean, you grow the cells, Then you subculture them.
Yes, sir.
So here we do something called splitting. What happens? Let's say our cells are grown. They are grown in a flask, so they will these cells will only adhere to the surface. Okay, they will not grow like bacterial cells do. Although there are some cells which can grow in suspension also, but that's not the topic here. So the cells, they only adhere to the surface of the if you will see those kind of flasks, they are called T25 flasks, T75 flasks. They have a surface that is coated with collagen, so the cells will only grow there. So they require this, they have some adhesion proteins, they require this adhesion, it is important for their cell growth. Once they, they are completely filled, like they are growing in the in one surface, once it is completely filled, we call this cell has reached its confluency. So the term confluency is clear to you?
Sir, I will.
So if you are growing a cell, what happens in bacterial cells? You see there is a growth curve, something like this. So there is a lag phase, then there is a log phase, and then there is a stationary phase, and then there is death phase, right?
Yes, sir.
So bacterial cells, usually you grow it in a suspension medium, like you in an LV broth, you grow it like that. Clear? I mean,
Yes,
is it
sir.
true or not?
Yes, sir. Yes, sir.
Yes, sir.
Sometimes you grow it in LB agar also, but we are not discussing about that. In this case, what happens? You use mediums like DMEM or RPMI. These kind of mediums are used. These are the media. These are animal cell culture media, very similar to LB broth, what you use for in case of bacterial culture. So you are growing the cells. The cells, they only grow on the surface of the flask that you are growing in. It doesn't grow anywhere. It doesn't grow in the media as a suspension. It only grows in the surface.
So why is it?
Why?
Why does it only grow on
That
the
is the
surface?
property of the cell. That is the property of the cell. I mean, there are reasons behind it. I don't recall everything, but this is how the cell grows, right? So they only grow on the surface. I will maybe clear it out in the next session. I'll have to prepare something for that. But what we do is we grow these cells, they reach confluency. Then we split the cells. So there are ratios. Let's say we use a 2:1 split ratio. So what we do here, we will centrifuge the cells. We will get the cell pellet, right? And we will divide this pellet, like half of it we will then go for subculture, and the rest half we may freeze them, like for storage, or we can even throw them out. That is not important. But only half of the cells we are growing for, I mean, we are only taking half of the cells for subculturing. So till this, the thing is clear.
Yes, sir.
Then again, what will happen? It will again reach confluency. Confluency is like maximum
Yes, sir.
amount of cells have, grown, and it
Okay, sir.
cannot grow any further. Then we will do again cell split. So this is how the cycle will continue. Like we do subculture in case of bacteria, there is splitting
What
in
is
case
this,
of
sir? What is this 2:1 split?
2:1 split is like half of these cells you will take for subculture. So if it is like, let's say, a 10:1 split, then 1/10 of the cell you will take for subculture.
Okay, sir. Okay, sir.
So it's basically the inoculum size in case of bacteria. That is what it is. So let's say you take half of the cells for subculture. How much time will it take to reach confluency? I mean, you are taking, let's say, half cells, and in one place you are taking 1/10 of the confluent cells, which will take longer to reach confluency.
The 1/10 portion.
Content.
That is correct. That is correct. Okay, fine. Now each cycle of grown and being split, so from here to here and before subculturing, that is called a passage. So we always identify these, animal cell cultures with their passage numbers.
Okay, sir.
That is clear. That is in case of bacterial culture, it is not important because bacterial cultures, they can grow and grow, that you can subculture them infinite times, but the same is not true for animal cell cultures. So that's why this number is very important, that you need to know how many times it has been subcultured, right? Is
Sir,
that
is
clear?
there any limit to means the passage number? It is
Yeah,
specific
that
for
is
every animal
what we are
cell.
going to study. That is Hayflick's limit.
Acha. Understood.
Okay. So see, now this is clear, I think, to most of you. Now we will talk something about population doubling. So what do you know? Have you heard about this term, population doubling?
Yes, sir.
What is it?
Uh, Population doubling is basically the phenomenon in which the total amount of biomass doubles in a certain period of time.
That is true. That is true. So let's say you have like half of the cells, half of, let's say, X. X is the number of confluent cells. Now it reaches X. And I mean the confluency level is achieved. So what is the number of population doublings in this entire event? How many times have the population doubled?
Uh, 2 times.
2 times or
No,
once?
One time.
no, once.
One time.
One time. Okay. Now you are taking 1/8 of this culture and you are now getting a confluency of X. I mean, X is the number of cells which gives you a confluent, culture. So how many times have the population doubled here?
3 times.
It's 2 times.
3.
2 times.
No, sir, 3 times.
Think
Eight.
again. Think again.
8 times.
1/8. Say what will happen if the population doubles? What if the population doubles?
1/4.
Okay. What will happen then if the population doubles?
Half X.
Half.
Acha, 4 times.
And then 4 times, no? It will be 3 times.
Yes.
1, 2, 3. We started with 1/8. Is that clear? Number of population doublings.
Yes, sir.
Okay, so let's see what Hayflick has seen. So in the right hand side, you see the doubling of these are fibroblast cells, WI38, as a function of their split ratio. So when they have, so they have given the number of splits and the number of doublings. So splits and doublings is clear. Splits is number of times they have subculture.
Yes,
And
sir.
doublings is what you have seen. So, okay, fine. So if they have done all 2:1 splits, so they have total number of splits that is required to achieve phase 3. Okay, what is phase 3 again? Let's go back. So this is a growth curve. Is it a growth curve? What will be here? Cell number.
Yes, sir. Cell
And
number.
x-axis.
Time.
So this phase is phase 1 that we call as the lag phase in case of bacterial culture. This is phase 2 and this is phase 3. Phase 3 is when? Now, how is it different from that of bacterial culture? I mean, why can't we call it as lag phase, log phase, and stationary phase? Let me tell you why. Because beyond this, the cell cannot divide. Even if you give enough of nutrients, enough of dividing, supplements, which is necessary for division, it will still not divide. The cell will die. That is what happens in a normal cell. Is that clear?
Yes, sir.
Now, what if the cell doesn't die?
cancer.
Yeah, you're right. You're right. So phase 1, phase 2, phase 3 is clear.
Yes, sir.
I'm not understanding if you are clear with all of this, but anyway, let's proceed. So number of splits have been 42 and the number of doublings has been 40. If we do 2:1 splits until passage 14 and then we go for 10:1 splits, so what will happen? The number of splits will decrease, but the number of doublings to which 57. Similarly, if we change the ratios so far, I mean so forth, the number of splitting will change. The number of splitting means the number of times your subculturing will change, but the number of doublings is somewhat close to 50 plus minus 10, right? Can I say that?
Yes, sir.
So what is constant of in a cell? The number of times you can split them or the number of population doublings?
Number of population doublings.
That is exactly Hayflick's theory. So that is what he studied. So he passaged different. I mean, he took a cell line, he cultured them serially. I mean, he subcultured them and he saw how long he can actually keep them alive. So till here, if you see till 50 passages, if you see this graph on y-axis, I mean on the left hand side. So till 50 passages, he could like make the cells survive. After that, even if he froze the cells, let's say like 10 passages he has cultured, then he froze the cell and then he again took this cell back, revived it. How many number of times will it uh, survive? How many passages? If 50 is the total number.
5. No, sorry, 4.
40, na? 40 plus 10, total will be 50.
50.
Is it clear or is it not?
Yes, sir.
The terminologies are a bit difficult for you guys to understand. I understand because you people are not very familiar with this. But anyway, the total number of passages, the total number of times you can subculture as let's say this particular cell is 50 times. Now you subcultured it 10 times, then you froze it, you take it back, revive it. Let's say you have kept it for 2-3 months and then you revived it. Then again, you can subculture in the same ratio for let's say 40 times. So it appears that the cell remembers how many times it has divided. Even if you store them, even if you change anything, it it tends to remember how many times it has divided and how many times it is left to divide. Is that clear?
Yes, sir.
So why do you think that happens? Any hypothesis, any theory?
Sir, please repeat the question. I just.
How do you think the cells they remember that how many times they have divided?
Sir, it might be. I don't know whether I'm correct, but it might be due to the fact that since normal cells, like the basic difference between normal cells and cancerous cells, one very basic difference is that of the telomerase activity. So
Right.
the thing is that when a cell is dividing, actually some part of the DNA is being lost because DNA polymerase cannot polymerize in the 3' to 5' direction. So some part of the DNA is lost in every replication cycle, and hence in every cell cycle, some part of the DNA gets lost. But so after so the total amount of DNA, so the total DNA, the time which will be required for vanishing of this total DNA is will be constant because the DNA size is also constant for one particular organism. But in the case of cancer cells, which does occur, that the telomerase activity remains active, so that's why they do not die. They
So I
remain
could say
active.
that the length of telomere is the way
Length of telomere has, yes sir. Length of telomere is a parameter.
We will read about these papers next week. You are right. This is how we assume. I mean, this is one of the hypotheses. Length of telomere is how the cells know how many times they have divided and how many times they can divide. So we will see how if we increase the amount of oxygen, if there is like a hyperoxic condition, so there will be more amount of ROS. It will cause a reduction in telomeres, the length of the telomeres. The cells will divide not that frequently. While if we somehow enhance the telomerase activity, then we observe that there is, potentially unlimited number of times a cell can divide. So limitless replicative potential was one of the hallmarks of cancer, if you remember. So yeah, that is still now we are having a fine difference between aging and cancer, how they are diametrically opposite to each other. Here also in the table 7 in the right hand side, what you see, uh, you see strains which have been split. I mean, they have been cultured, they have been split in 2:1 ratio. Some of the cells, they have never been preserved. Some of the cells have been preserved. So what we essentially observe, no matter whether you preserve these cells or not, the number of times, the number of cell doublings they can undergo is constant.
Yes sir.
Is that clear?
Yes sir.
Okay, fine. I think that's the end of the day. I mean, I'm not going to too much detail today, since one of you already said it's already too confusing. Anything else? I'll just stop the recording here. Just give me a moment.